So today I'll discuss why R. Okay. Next part is I'll describe about the processes of clinical trial. Okay, what are the different processes, protocol development, and other. Okay, then we'll move to the role and responsibility of the clinical R programmer, and at the very end we'll have a question answer session. Okay, so this is the agenda for today. So let's jump. And why R? So as you all know, right, R is a free and open source. Okay, so we don't have to pay anybody any pennies, right? You can download and you can install in your system, and you can do whatever you want to do with the R. Okay, so these are the advantages we have, right? And it is having a large community of users. Okay, and they are creating various packages, various tools. Okay, and we can very easily utilize for our day-to-day job. Not the third part is it has an independent of platform. That means we don't need a very sophisticated configuration of laptop, right? We can install R in any normal with a basic configuration, and it runs across all platforms, whether it is a Windows, Mac, Linux, right? It has a presence across all operating system, and it also has a presence on a cloud-based computing part. Okay, and the last is this is a gateway of a lucrative career. So right now there is a huge demand across different pharma companies related to our programmer. So there are opportunity. We can just train ourselves and we can utilize the opportunity right, that is out there. So these are the important features, right, we have in R. Okay, I'll show you a growth of R. So here, this is a slightly old figure, right? But it can give an idea, right, about the growth. So on the y-axis, we have percentage of job posting, right? Percent of matching job posting. And then here in the x-axis, we have years. And we have three line diagram. So one is the orange one. Orange is for the SAS statistics. Blue one is for R statistics, and green is for Python. And if you notice, from 2015 onwards, so R basically speaking, right, and then in mid of 2016, it basically crossed Python as well as SAS. Okay, so this is the opportunity, right? And in recent years, there are huge demand across pharma and other industries. For example, in data science, in media marketing. Okay, for R. So I'm just emphasizing here for the clinical trial. Okay, so you can very easily do all kinds of things what we generally do using SAS and other software in a clinical trial. So we just have to use R, and we have all the packages, right? We can do everything, right, with R without relying on any other software. Let me cover one more other this bar diagram, the histogram basically. And here, if you notice, this is basically a segregation of use of R, right, or traffic in different industries. So you can notice the academics has a very large number of traffics. This is basically having 7%. Healthcare, this is near about yeah, so this is around 8. So academics is having 8 point something. This is healthcare is 6 point something, right? Then government, then consulting, insurance, energy, right? So mostly the traffic is coming from these two industries, mainly academics and healthcare. But we also have presence in other industries, right? Like for example, finance, retail, media marketing, right? You can notice here manufacturing, tech, and electronics. Here in the consulting, this is also very important because here these are open source. You don't have to buy for any subscription. Okay, so you can do consulting with R, right, without relying any paid software. So these are the potential we have, right? And this is a high time we can very easily train ourselves, and we can get the opportunity that is present out there, okay, in different industry. So in the healthcare, quite almost almost equal to academic, right? And yeah, this is data from Jan to August 2017 in UK and US. Okay, so I'll pause here. Any question, anybody?
No, Ranjit, we are good to go.
Okay, perfect. So the next is here, I'll describe briefly about the clinical trial processes. Okay, so first is the trial protocol development. Okay, this is at the very early stage. So the different stakeholder. This is not the responsibility of the programmer, right, or a biostat or a clinical team. Mainly this derive right. This is a role and responsibility of the clinical team, but this is a kind of collaborative effort. All different line function contributes to a trial protocol design stage, and this is a bible, a basic documents for a clinical trial. So let me show you a protocol. So I'll show you the protocol of our studies, right, which we'll describe in the part 2 of the course. So here we have a protocol. Yeah, and this is a double-blind placebo-controlled study to assess the efficacy and safety of active drug versus placebo in the patient with mild to moderate COVID-19 disease. So there are a whole lot of things here. Okay, let me highlight a few parts. So first, I already highlighted this is a disease condition. Yeah, this is our disease condition. Give me a second. Yeah, so this is a disease condition for which the company is trying to develop a medication. Okay, and here we are just targeting mild to moderate patients, right, of COVID-19. So we are not targeting all mild, moderate, severe. Basically, we have three kinds of patients, right? So they are targeting only the mild to moderate. Why not severe? Because severe patients may have other health related issues. Okay, and despite having a very good or efficacious drug, they're not able to basically approve the efficacy. Okay, so that's why companies is they basically decide these in a very strategic way. Okay, and that's why they have not included a severe. This is just another perspective. Okay, the most important part is here the double blind placebo control study, right? So let me just highlight the other part and then I'll just revisit this one here. Okay, so here this study is basically to assess the efficacy and safety of oral active drug versus placebo. So the company is trying to develop an oral active drug, triple A. This is the molecule name with the placebo. Okay, so this is the thing. So they just want to test the effectiveness or efficacy aspects and safety. Okay, so efficacy is related to effectiveness of anything, and safety is basically whether the molecule is safe enough, right, or not. So this is the main part here. This is a phase 2 studies, so that's why they are targeting efficacy in as a primary input. I'll cover the primary endpoint in a minute. Okay, and here they are comparing with the placebo. So first of all, why we need the placebo? Why we have to compare with the placebo? Because placebo can provide idea about the references, right? Because if you don't give medication to a patients, they also some patients shows improvement, right? They basically improve over time, right? And they become better. So because of that, we'll have to also assign some patients to a fake pill or a placebo so that we can get an idea about how many patients are recovering without any medications. And that provides some kinds of reference or maybe a label where we can compare the test drug with the placebo so that we can get an idea about the true treatment effect. Okay. So the job is here just to get an idea about the true treatment effect. Okay, give me a second. True treatment effect. Yeah, so this is the main part why we generally include placebo in the studies to compare with the test and get an idea about the true benefit of a molecule or a drug. Okay. The next part is here, that is double blind placebo control study. So again, this is placebo control is basically a placebo. Why? Because the company want to compare, or the sponsor want to compare a true beneficial effect of a molecule. And why they have to do a double blind. Blinding is very important aspects in a clinical trial, because if the patient knows that he or she is taking medications, right, and if other patient knows that he or she is taking a placebo, they behave in a different way. Okay. If the patients if the patient knows that he or she is taking medication, the actual drug, so they feel better, right? And this is called a kind of placebo effect. Okay, just to avoid that part, right? Or just to avoid those biases that patient may create. Okay, we generally blind. Okay, so we just devise some kinds of operational procedure so that nobody knows whether they are taking a medications or a placebo arm. So here we have had three kinds of blinding: single blind. So in case of single blind, only patients is blind. Okay. So what they do, they try to devise some kinds of operational procedure. For example, if you have a placebo drug and active drug, what they do, for example, this is active drugs. This is the size of active. This is in red pill, and placebo can be of a green color circle. Okay. So what they devise, they try to introduce a kind of a medication, right? uh, in circle. Let me use the same color as of, yeah. So they create again a circle, and this is a molecule, this is a active drug. Okay. And in the case of placebo, that is in green color, right, they also create some placebo with the triangle, right, red triangle. So here this is a placebo. So patient cannot distinguish whether they are taking medications or a placebo, because they can get this red one, or they can also get a green one. Okay, and green one is for active drug. Here green one is for a placebo. So this is the way just to blind patients. There might be other connotations, right? So a patient is blind, this is okay, but there might be the case the investigator, if the investigator knows that a particular patient is taking medications, or different patient is taking, the other patient is taking placebo, so he or she may assess the result differently. Okay, so we also have to devise some way just to blind the investigator. So what we can do, we can recruit additional investigator. So one investigator basically administer the drug, the other investigator measure the assessment. Okay, so in that case we can blind also the patients and investigator together, and that is a kind of a second or double blind, right? So double blind here, we blind patients and investigator. Then we have also a third triple blind, where we blind all stakeholders that are involved in a clinical trial. Okay, so if we increase the level of blinding, it basically incur the cost. So that's why company just decide this very strategically, and that's why here we have double blind. Okay, so this is all about the trial protocol, right? Protocol type. I'll just give you a other idea, right, about this protocol in a minute, right? So I'll pause here. Any question, anybody? Hope this is clear to everyone. Don't hesitate if you have question.
No questions from me.
Okay, okay, okay. Thank you. Then I'll just move to the next part. So if I go further down, so here we have this page. Yeah, we have this table of content, abbreviation, and then here we have a protocol synopsis. So synopsis is basically a kind of summary, right, about the protocol. And here you can notice we have the trial protocol title. Again, the same thing. This is a protocol number, then the sponsor name, then the CRO name. Okay, and here we have a study site. So multiple sites in USA and elsewhere as appropriate. So what the site means? A site is basically, it can be healthcare center. It can be primary healthcare center, secondary healthcare center, tertiary healthcare centers, and it can present in any locations. For example, it can be globally. For example, some trial in India, some trial in China, some trial in USA, right? Some trial in UK. So there are different advantages and also disadvantages of including patients from a different region. Okay, and the healthcare centers, right? It can vary from primary healthcare centers to a tertiary healthcare centers. For example, if we are targeting a molecule, right, with a very sophisticated disease condition, for example, cancer stage 2 or stage 3, so in that case the patient's condition is very critical, right, and the patients basically we need to recruit the patient from the hospitals. Okay, but in case of COVID-19, right, this is not a problematic part, right, or maybe other kinds of fever or maybe diabetes patients. Okay, so it depends, and accordingly we can recruit the patient from a home also, right, if that is related to mild pain, right, and other disease conditions. So the sites can be primary, secondary, and tertiary healthcare centers. Okay, so and then it can be present in any locations, right? It can. Yeah, so I'll give you one idea. So right now in this study, we have multiple sites, but all the sites in USA. We don't have sites from other region. So here the sponsor is trying to create a kind of study design, right? Trying to take the patients from USA. So sometimes the patient, the maybe some other studies, right? They may have patients from a different region. So the benefit of including patients from different region is you don't have to do additional studies, right, for the approval. You can, for example, if you are trying studies A, right, here we have patients from USA, UK, Asia, Africa. So what is the advantages? So we can very easily recruit all the patients from different regions, and we can file the new drug applications in all four regions simultaneously, just considering the data of a study. Okay, you don't have to do additional studies. So this is the benefit. You can get the approval for a particular region just considering the same data sets. The disadvantage here is, so there might be the case the conditions or maybe it can introduce bias, it can introduce a whole lot of spread or variability. What variability means here? So for example, the climatic condition of USA is very different from Africa. Okay, So this may also contribute, right, somewhat for the in the diseases conditions, right, and it helps, or maybe it helps in a positive way or it helps in a negative way for reducing that disease condition. For example, in the case of psoriasis study, right, climatic condition is one of the factor that basically influence the treatment effect. Okay, so if we have, if we are doing maybe kind of psoriasis study, so in that case you'll have to be very careful about the region, right? But for others you can choose, but also there are other factors, for example food culture, right, food habit, sanitation, level of sanitation, level of education, these are also contributor, right? For example, if you're doing any kinds of infections study, right, so in that case the sanitation is one of the important contributor. Okay, if you are doing a study related to stomach, right, so food habit is one of the thing. Okay, some people prefer very spicy and other kinds of food, some are very raw, so these are also a kind of it can introduce a kind of heterogeneity, some having whole lot of variability, right, some are very stable, right, and if we have different kinds of data with different spread or different variability, so in that case you cannot able to get an idea about the you cannot measure the treatment effect correctly because of the heterogeneity or because of spread. Okay, so you can have here the benefit is uh, get an approval with the same data. Yeah, same data. And the disadvantage is this is a kind of plus point, right? The negative point is it introduce spread or variability or heterogeneity. So if you notice, most of the trial have patients from different regions. So how they basically curb or how they basically resolve the issue of heterogeneity. So for that, they try to introduce inclusion exclusion criteria, right? So that they can have a same patient profile, right, in the test arm and also in the placebo arm. Okay, because here in our study we have test arm and also the placebo arm. Okay, so here we should have some patient population, here also we should have some patient population. Okay, so we we should advise some kinds of uh, technique so that we should have similar kinds of patients, so that whatever the difference we have, that is due to a test drug and placebo. There should not be any other factor that creates the differences, because whenever you are trying to measure the treatment effect, the other factor contributes and you cannot have an idea about the true treatment effect or true benefit effect. Okay, so that's why we should have inclusion exclusion criteria. I'll come to that here. We should have at the bottom side. Let me go there and describe some part. Yeah, so here we should have inclusion exclusion criteria, and these are very general here. This is not a very sophisticated one, and this is related to COVID-19. So here we have RT-PCR test. Okay, you can notice here. Okay, so here we have RT-PCR test. So RT-PCR test uh, is just to diagnose the COVID-19 symptoms, right? Or just to confirm that the patient is having a certain kinds of certain symptoms or a certain strength, right? Mild, moderate, severe, right? Because we are just considering the patient with mild and moderate. Yeah. So that's why they just have to go through this diagnosis. and the other are very simple one. So they just have to confirm, right? Just the patients has to satisfy these eligibility criteria, right? Inclusion criteria, and some are exclusion also, right? So it should not be a breastfeeding woman, G4 PD deficiency. This is because this can hinder, right? It can create differences in COVID-19 cases. So that's why we just have to include the patients having these kinds of deficiencies. Then other psychotic disorder and current psychotic symptoms, because if the patient's having some kinds of psychotic disorder, so they cannot provide a feedback correctly. Okay, so that's why we generally include this in most of the studies. And there are other things, right? So because they don't want to capture the other disease conditions so that it can create difference other than the treatment and the placebo. Okay, so patients basically visit the site, and so the investigator, right, or maybe a nurse, right, they just have to check all the inclusion criteria and exclusion criteria. This is in totality, right, we call eligibility criteria. So patients should satisfy all of the inclusion criteria and none of the exclusion criteria. Okay, so if the patient satisfy all of the inclusion and none of the exclusion criteria, then only the investigator recruit that patient in the site. Okay, if any patient satisfy any one of the exclusion criteria, so in that case they can not include that patient into the study. Yeah, this inclusion criteria and exclusion criteria, this is devised just to make the patients homogeneous. Yeah, or minimize variability. So this is the part. So let me go to the top and then I'll describe the other section. Okay, so I'll pause here. Any question, anybody so far? No question?
No, Rajit.
Yeah.
Yeah, it's good to go.
Okay. Okay. Thank you. Yeah. So here we have phases. So study phase. This is phase two study. Okay. Then the objective. This is very important. So at the very end, right? So the any protocol have some objective that the company want to achieve. Okay. And here the objective is you can have text. Objective is to determine if the active drug trademark this one and increases the proportion of patients with clinical recovery. So this is very important. Increases the patient proportion of patient with clinical recovery from COVID-19 symptoms on day 14 in the patient with mild to moderate COVID-19 disease compared with the placebo. So the text that I highlight, this is very important. So the drug that the company is trying to develop basically increases, right? Uh, their aim is just to prove that the drug increases the clinical recovery at clinical recovery on the symptoms of COVID-19 on day 14. So first of all, why they are measuring at day 14 only? Why not day 7? Because there might be some clinical justification or clinical studies that may satisfy that if the patient shows clinical recovery on day 14, that is considered stable. Okay, so that's why they are choosing this day 14 as a cutoff. Okay, and what is this clinical recovery defined, right? So here we have a clinical recovery. So if the patient shows all of this any one of these criteria, right, that basically is clinical recovery. Okay, so temperature should be less than 37.7 degrees centigrade, respiratory rate should be less than 24, shortness of breath should be absent, cough should be mild or absent on the patient reported scale. And you can recall in the COVID-19 time, right, we generally have these kinds of symptoms, right, and we try to just get an right of resolution based on this parameter. So this is the thing. So if the patient shows any one of the criteria, so that is considered clinical recovery. But one thing is very important: we need to assess that these clinical recovery aspects on day 14 only. So the patient took medications, and on day 14 we do have to do the assessment of a clinical recovery endpoint. Okay. The next part is along with the primary objective, right? We also have to have some other thing that the companies want to explore, okay, and those are under a secondary objective. Because the primary objective is the main objective. Based on that, we should have a primary endpoint, and based on that, we'll have to have sample size calculation, power calculation, right? So this is very important. This is our main part. But the secondary, these are basically sometimes powered or non-powered. So what powered means? So powered means we have sufficient number of subjects, and at the sample size as power power calculation stage, the statistician or biostatistician basically consider sufficient number of subjects so that they can test the primary objective as well as secondary objective. But this is very less, because if we want to test multiple hypotheses, right, or multiple endpoints or multiple objectives, they need to use a large number of subjects, and including large numbers of subjects incur cost, right? So these are the other part. But sometimes if this is required, so the companies decide to recruit additional subjects so that this primary objective and secondary objectives both can be tested simultaneously. Okay, but these are very rare in most of the studies. So the company is trying to test this primary objective, and secondary is just for exploration part, right, or some additional benefit that they want to establish with that molecule, so that they can market their drug right, as compared to competitor in a better way. Okay, so here we have the secondary object. This is a decrease in hospitalization rate due to COVID-19 on by day 14, decrease the number of medical follow-up, then increase the proportion of patient with absence of clinical symptoms, and right. So these are again another kind of measurement, right, that shows a recovery from a COVID-19 symptoms. Okay, apart from the secondary endpoint, so here we have a dosing day, right? I'll skip this part. So these are the objectives. Objective is basically a clinical questions, right? So everything is written in a clinical way, right? But we should have some measurement, right? Very quantitative measurement, so that the team can write a code and they can validate, right? They can basically interpret whether we are able to achieve this primary objective or not. So for each objective, we should have an endpoint. And here, if you notice, we have endpoints here. This is primary endpoint, and this is a very crude or maybe basic level of questions. So proportion of patients with clinical recovery. So proportion means, for example, here we may have patients, 100 patients. Out of 100 patients, some patients shows clinical recovery, so this is yes. Some patients shows clinical recovery, that is no. And then we should have to count these categories, right? So we can count the recovered patients and non-recovered patients, and here we'll have to divide this count by total to get an proportion. Okay, this is a simple thing. So this is a primary. So primary endpoints is basically a measure, a statistical measure or quantitative measure that provides an justifications, right, for a objective, for a primary objective. Similarly, for the secondary objective, we should have secondary endpoint, and secondary endpoint are basically a measure that quantify all the secondary objectives. So if you recall, here we have multiple secondary objectives: decrease in hospitalization rate, decrease in number of medical follow-up, right? And here we have, so this is hospitalization rate. So rate is a measure that can provide an idea about the secondary endpoints, one of the secondary endpoints. And here number of COVID related medical follow-ups. Okay, if we have a less number of medical follow-up, that means the patient's condition is getting better. Then also proportion of patients with COVID-19 symptoms at day 14 by individual symptom assessment. Yeah, this is again one endpoints. Then we should also have here, we have exploratory endpoint. This is for exploration, right? So that the company can get an idea about the these aspects, and they can devise a new trial. Okay, so this is basically for exploration purposes, and based on that they can design new trial. Okay, so these are the endpoints we have under the secondary. Time to maximum severity of COVID-19 symptoms after the start of treatment. Then this is a time to endpoint. Then time to resolution. Then again viral load, right? Because this is viral load is one of the indicator, right? That shows an idea about the infection. Apart from all the efficacy endpoints, we also have to establish the safety, because if the molecule is not safe, you cannot think of efficacy, right? So that's why we have here a safety endpoints, and here we have to validate these aspects: the adverse event, right, a vital sign, and a chemistry and hematology label of parameter. Okay, then here the drug administration. So the drug is self-administered, right? And this is a tablet size and strength, right? And then the drug is given on day 1, day 2, day 3, and day 10. So we have to this is the schedule of a drug. Okay, and then we'll have to do assessments, follow up, right, on day 14, just to analyze the primary endpoint, and based on that we can get an idea about the approval, right, whether we can get an approval or benefit, true benefit rate. So that I'll show you in the part 2 of the course, right? I'll just revisit this again, and then I'll create the tables of primary endpoints, secondary endpoints, and also the adverse event, like vital sign, right, disposition, then demographic, and all tables one by one. So these are the aspects, and here this is very important. So number of planned subjects. So they are approximately planning to 175, but they recruit 250 randomized in the studies, and this 250 is for the segregated, right, split, half in a test drug and half in placebo. So 125 in triple A, and then 125 in placebo. Okay, so these are the thing here. Let me check, is there any other thing? And here this is the randomization procedure. This 1:1 ratio means total patients we need to segregate in 2 parts, right? So this is a treatment group, triple A and placebo, and at least 250 subjects. So half, that means 150 in arm A and 150 in placebo. Okay, and then here we have to go through the screening procedure. This is not our responsibility. This is basically for a clinician or investigator that is present at the site. Okay, they just have to check all these criteria, and if they think that these are okay, right, so they only recruit the patient into the study. Okay, so let me go to the next assessment, next part. So this is again a efficacy assessment, right, in detail. Then here safety assessment, then statistical aspects, right, statistical analysis. So here we have a definition of ITT, safety, PP. So I'll discuss that part when I do the part 2 of the course, right? So I'll just revisit this part. This is very general. So ITT is one species randomized, so we need to include that into the studies. Why? Because if you do the randomization is basically a scientific process, right, or scientific technique that basically justify us that everything is of a same, every patient is of a same characteristics. Okay, so that's why we cannot exclude the patients, right, all the patients randomized. So once patient randomized, we need to include. Why? Because it can provide a justification that everything is balanced. Okay, there should not be any difference between the patients group, the group that is taking the medications, and the group that is taking the placebo. There is only one difference, and that difference is due to a test drug and a placebo drug. Okay, so this is the main part, because this randomization here we generally assign treatment and placebo, right, randomly. Okay, and because of that there will not be any pattern, right? to the patients. So that's why in ITT, most of the time, if you notice, all randomized patient we need to include, and all the efficacy related assessments we generally do on the ITT population, because these are randomized population and every baseline characteristic is balanced, right? And, that's why we devise this population in all primary and secondary endpoints. Then we should have safety. So there might be the case the patient randomized, but they didn't receive the medications. So if the patient received the medication, then only we need to include the subjects into the safety population. So here, patient randomized and receive at least one dose. Why this is called safety? Because if the patient is taking any medications, so it is the responsibility of the sponsor company just to make sure that the patient does not have any kinds of serious consequences, right? So that's why they just have to include into the safety population, right? And they have to do a thorough review, or they just have to keep those patients in a very controlled settings, so that if anything untoward happens, right, they can provide different medications and switch to another treatment arm or withdraw or whatever. Okay, so that's why we have the name safety, because it is the responsibility of the sponsor company just to make sure that particular medication that the patient is taking, right, is safe. Okay, because at the very early stage, nobody knows much about the mechanism of working of a particular molecule, right? So anything can go wrong at any point of time. So that's why they just have to do maybe a very close, close review, or close just have to put into a very controlled settings, right? So that they can provide any kinds of therapeutic help, right, in case of emergency and other thing. Then we should have also another population. This is called per protocol populations. So there might be the case that during the clinical trial settings, we may have to take medication on so and so day, right? But the patients forget to take medications, right? So these are kinds of violations. So violation can happen during the course of trial, right? And it is a responsibility of the clinical team just to categorize those violations in the form of mild and minor and major. Okay, and all the major that basically provides some kinds of data integrity issues. Okay, so in that case, we need to exclude those patients from the population group, right? And we need to only include the patients who are very compliant and does not have a major violations. Okay, and this per protocol patients, these are very clean patients. Okay, satisfy all the criteria as per the protocol. Okay, so this is the aspects. Okay, so I'll pause here. Any question, anybody? So this is all about the protocol, right? Here we have the other information about the molecule, right? Then here the chemical structure, okay, non-clinical studies, right? So at the very beginning, the company has to do non-clinical studies. Non-clinical studies are basically study done on the animals, right? And this is just to study the toxicity level, right, of a molecule. Okay, it can have genotoxicity, it can have carcinogenicity, right? And after that, right, they have to recruit the healthy volunteer, right, and then so on. So I recommend maybe you just go through these aspects, right, and then we'll repeat this protocol once again in the part 2 of the course. So I'll just go through here. This is a schedule. This gives us an idea about when we are doing which kinds of assessments. So informed consent at screening, then the other part, right, then other kinds of things. Here, if you notice in the screening, we have minus day -4 to -1. Why we have minus? Because day 1 is a randomization. This is a day of randomization. Okay, so before that, we'll have to go through a whole lot of screening procedure, and whole lot of screening procedure we cannot do in 1 or 2 days. So that's why we should have a buffer, right? And that buffer is represented in minds. Okay, so this is the mind thing. Okay, so I'll now jump to the slide deck. So first part is the preparation of a trial protocol. This is a document that is basically maintained by a clinical team, but this is a collaboration of a different line function, right, or different stakeholder. After the trial protocol once we have a trial protocol finalized, so then we have to do the site selection. Site selection is same selection of hospitals or healthcare centers, so it can be primary, secondary, tertiary healthcare centers based on the disease condition that we are targeting. After the site selection, and also at the site selection stage, right, companies, maybe big pharma companies, have their own list of sites or hospitals. Okay, they do some internal studies, futility kinds of studies. What this study is meant for, these are basically a study that is done that which site has provided a very very good quality of data earlier, right? So they have their scores, right, for each hospitals, right, and they basically have a lower score, right, for these hospitals that don't provide data in a clean manner, right, and based on that they choose the site. Okay, so sometimes they do some kinds of ad hoc studies, right, just to get an idea about which hospitals is good, which hospitals have facilities. For example, if the hospitals does not have a certain kinds of ward, right, it is not required, right, to include the patient from that hospitals. So hospitals may have sufficient number of facilities, right, and maybe because in case of sophisticated disease, specific kinds of instrument or specific kinds of laboratory is required, right. So these are the aspects. And then we'll have to go for the approval processes, right. So all the trial protocol, right, we need to submit to our maybe professional health professionals or patient advocacy group, right. These are the person that is not involved in a clinical trial, and they just check these protocol, right, for any kinds of maybe potential risk. Okay, they suggest some kinds of advices just to modify or just do some kinds of modification in the protocol. It is basically mainly a sponsor call just to accept or not, right, but on the good aspects, right, they basically accept all the suggestion that they receive from approval processes. Okay, once the protocol get approved, so then the patient recruitment start, and at the site the clinicians, or sorry, investigator or nurses, they basically check all the inclusion exclusion criteria. If the patient satisfy all of the inclusion and none of the exclusion criteria, then they recruit those patients into the study. And then the CRF part starts. CRF is nothing but a case report form. This is a paper document, right, where the investigator records all the values. Okay, sometimes it can be a paper document. Nowadays we may have eCRF, electronic version of that, right. So just writing, right, instead of writing onto the A4 paper, they just use some devices. It can be mobile phone, it can be tablet, it can be laptop, right, and they record all the measurement on that device. The benefit of having eCRF, electronic CRF, is we can get the data on a real-time basis. Okay, in case of CRF, they just write on a paper document. After that, they just have to send this CRF to the data entry team. The data entry team enters all the values into the database, and then we should have another team just to validate those records, right. So this is the process, right, but right now most of the time, right, we should have an electronic trial, yeah, electronic eCRF, right, to record the data. After the data entry and validations, they basically dump the data to a database. Okay, and then all the stakeholders, whether it is a statistician, clinical team, programmer, all have the access of the database. We generally check the queries. Okay, there are a whole lot of things done, right, during the data cleaning aspects, data cleaning stage, right. And then if every person is okay, right, the data is clean now, right, they can go for a database log. So it can have soft log, hard log, right, it depends on the analysis and it depends on the stage of the studies. And once database is log, right, so here we have to get the database and we have to do the analysis. So we have to write the script, and then we can do the report creation. Okay, so before the database lock also we have the access, and the different or separate team basically writes the program, right, for creating a report. And then we'll have to submit this report to the result meeting, right. These are basically a different line function. Okay, they just check all the reports, and if they need that some extra report is required, so they can suggest us to create the extra report, and then they just file those results to the regulator, okay, for the approval processes. And here I just use FDA as a notation purposes. It can be any regulator, okay. And then they have to submit to the FDA, and FDA go through all the thorough review, right. If they satisfy that everything is okay, so they can approve, otherwise they can disapprove that particular molecule. Okay, so this is a holistic process of a clinical trial. I'll pause here. Any question, anybody?
No, Ranjit, you are good to go.
Okay, so this is a kind of a very descriptive part, right? So maybe tomorrow we'll have another class for a history of and development of our, right. So this is again a history part, describing all the development stage. But on class 4, right, we should have a programming. So on class 4 I'll just install the R and R studio, right. So sometimes descriptive is very boring, right, but I recommend this is very important. And sometimes the interview process they ask, right, these kinds of questions. Okay, so I recommend just to have some attention, right, and don't hesitate. If you have any question, you can ask me. I'm here just to help. Okay, so next part is here the role and responsibility of the clinical R programmer, right? So here at the very senior level, so we'll have to just maybe collaborate for a trial protocol stage, and our collaboration is mainly for a data capturing aspects, right? How we are capturing the data, and maybe it depends also on this on the company, right? Whether we are working for a big pharma or a small CRO. Okay, in the small CRO we don't have a whole lot of resources, so we'll have to collaborate with the clinical team to get an idea about how they are capturing and how they are designing the CRF. Okay, so this is mainly role responsible, role related to a data capturing aspects. Okay, in most of the companies in the big CRO, they have a big pharma company, they have a dedicated team for CRF designing. Okay, and we don't have to collaborate, right. They can very easily do all kinds of their review, whether we have all the data captured in the CRF or not, right, as per the protocol. And we don't have to collaborate. But in case of a small CRO, we'll have to also collaborate just to for the CRF design stage. We generally have to review, right, whether we have all the data captured here, right, in the paper document or in electronic document or not. Okay, and sometimes we have eCRF. This is again electronic version of the same. So here also we have to review, right, because we make sure that everything that is mentioned in the protocol is captured, right, in the CRF or eCRF part. After the eCRF, this is our job, just to annotate the CRF. Annotation is basically writing some kinds of note there. Let me check where we have. Yeah, here we have annotated CRF. So annotation is kind of specifying variable name, right, and right, for a certain, for example, this is adverse event, and here we have the data system date of assessment. So here we'll have to use this one. This is for system date. So at adverse event description, this is AE AED AE term. So this is not a CDC standard, okay, but this term is mostly regionally used, sometimes from the CDC standard, sometimes the other companies. But right now, nowadays, all companies uses a CDC standard. So we have here a CDC guideline, SDTM IG, right? Based on this guideline, we'll have to annotate those variables, right? So let me go to the adverse event. Okay, let me use this way. A.xpt. Yeah, so we have here all the variable mentioned, right? And based on that, the company, we have to code. I'll just revisit this ASDM implementation guideline again, right, in the part 2 of the course, and I'll show you how to map those variables, right? So they basically use it is okay. Okay, let me open that. Yeah, so here, so this is our job, right? Just to annotate, keeping SDTM implementation guide as a document, right? Sometimes we may have to annotate, right, without following that SDTM implementation guide, but this is based on the requirement. But in most of the time, right, we have to follow the SDTM implementation guide, and we'll have to annotate. So annotation is just writing some very small name, right, along with the dataset. This is a dataset name and or domain name, and this is the variable name. Okay, because in every programming we cannot have this label, right, this whole thing as a variable name. Okay, so this is the maybe a programmatical part, right? And the annotation also helps in identifying the variable name, right? Because we have to standardize the things. Okay, so it can do two things. It can standardize the variable name, it can standardize the data, and also and the programmatical aspects, right? We cannot have a very big as a very big label as a variable name, so that's why we'll have to use a small one. So this is a programmatical aspect. So this is our responsibility to just to annotate all the CRF, right? And in most of the time, basically senior people just do, right? And as a junior level, we just have to create the data set. Okay, so now I'll move to the other part. Once we have annotation, right? When we completed the annotations, so we'll have to create the SDTM datasets. Just checking, just following the SDTM implementation guideline. Okay, this is nothing but the standard data tabulation method. So just mapping the similar kinds of data in a particular domain. Okay, we have a domain for DM datasets, A datasets, SV datasets, TA domain, right? So we'll revisit this part again in the part 2 of the course. Once we have SDTM ready, so we'll basically create the XPT file and we'll submit those data to the health authorities. After creation of SDTM, we'll have to use this SDTM as a reference, right, or a source data, and we'll transform into ADaM. ADaM is just for analysis data. Okay, so here in the case of SDTM, we cannot do any kinds of imputations. We cannot do any kinds of derivation. But ADaM is basically for data analysis aspects, and keeping all the endpoints in mind, they basically create the data, so that they just have to run a procedure or a function just to tabulate or count the data. Okay, so this is a main part, and once SDTM is ready, so they have to also submit that part to the regulator, and they also have to use this ADaM datasets for the analysis purposes. So they create table listing figure. So this is our responsibility. We'll have to write the code and we'll have to create all the table listing figure that is mentioned in the TFL cell document. Okay, and then we'll have to submit these to the health authorities for the approval part. So here, this is our responsibility: annotation of CRF, creation of SDTM datasets, creation of ADaMs, creation of TFL cell, right? So here is our job profile, right? We'll have to do these kinds of things if we join as our programmer. Okay, any question on this?
Ranjit, here you said that in the annotation we just give a words. Okay, some keywords might be which is which explains about that condition. Suppose
Correct.
in that you have taken one example, AE dot something else. AE is resembles that adverse event of something.
Correct.
So yeah.
Let me
In
show.
a first glance, this is the first time which I am seeing. So, how can you explain a bit more about that one and regarding SDTM, how it looks like actually?
Yeah,
That
yeah.
just wanted to know.
Correct. Yeah. So let me open that IG. Yeah. So if I go to the first page where we have all the domains defined. Okay, where it is. Yeah. Give me a second. I'll just.
Yeah.
I just want to. We have a page where we have all the domain mentioned, right? With categorization. Yeah, here. So the purpose of creating SDTM is just to split the data in a respective domain. Okay, so we have different categories: a special purpose domain. Then we have here the intervention. So a special purpose domain. So here DM is one domain that we'll have to create, and which variable we have to map here? All the variables that are related to a demographic. Okay, so let me show you what are the variable we have. dm.xbt. Where it is. Yeah, so here notice this is demographic, and here we have a list of variable. So we have to here variable name, right? Variable label, subject ID, domain use subject ID, then again RFS addresses. So these are the variable that we can only create. We cannot add any other variable, right? That are not mentioned here. Okay, so we'll have to create only these number of variable. And also here, if you notice, we have role, right? That is defined at the very end, the core part. So sometimes we have required. This is very much required. We cannot skip that. Sometimes we have expected. So we can have to we'll have to have variable name, but this variable can be blank. Okay. Sometimes we have permissible kinds of role, core. So this permissible. So if the raw dataset has this information, so we can map. Otherwise, we can drop the variable, right? And that is okay. So we can drop the variable if that variable is not there in the raw dataset in case of permissible variable. If the variable is required, anyhow we have to map, and we should have also the values. The variable cannot be blank. In case of expected, so variable name should be there, but it can be blank. So these are the list of exhaustive variable and that we have to create in case of a DM dataset. Okay, similarly we have other domain, for example AE. So in that case, all the, and if you notice the variable here, these are study ID, domain, use subject ID, subject identifier, right? Then here reference date, as subject reference, in date, right? Statement start date and all, right? And if you notice the variable like death date, participation date, informed consent date, then here death flag. So, and this is the basic demographic: birth date, age, hue, race, sex, ethnicity, and other, right? So because the name suggests that these are basically having a demographic, right? Age, sex, race, ethnicity, birth rate, right? Treatment arm related informations, okay? Then participation date. So these are the variable we need to map in the DM domain. Similarly, in the case of AE, where it is, let me just highlight that. So other domain, for example, comments, all comments that are mentioned in the trial, we'll have to map here. Subject visit. So for example, in the studies we have 20 visits, right? So all 20 visits we'll have to create, and we'll have to map to a subject visit. So how many subjects have achieved a kind of a visit, right? So we'll have to have subject number, then respective visit start date, visit end date, okay? Then intervention. So here, any intervention, any extra or any other substances that the patient is taking, those are a part of intervention. So here we can have exposure, we can have procedure, any kinds of surgery or radiotherapy or chemotherapy, those are a part of procedure. Then substance use, for example, patient can take any additional substance, for example tobacco, liquor, right, nicotine and other kind of thing. So we'll have to map under the substance use. So here we have different kinds of different buckets that captures a specific type of information. And why they have created this? Because if you want to know about the basic demographic about the patients, you can just open the demographic and you get an idea. Okay, so this is basically categorizing the raw data sets in a different way so that we can get an idea about a particular information, right, easily. Okay, so this is basically just to harmonize the thing. Okay, because in
understand.
in the company A they can have all the demographic page, right? And sometimes we also have substance use in the same page, right? So, but in the case of SDTM creation, we'll have to segregate those information in two different domains. Okay.
Okay, so this step, whatever you are talking, so whatever we get the data from the clinical trials, we are annotating. So we are just giving some names according to our SDTM guidelines.
Guideline, correct? Yeah.
Oh, yeah. Oh, good. So post this, okay, we will annotate. We have annotated, and after this, what will happen in the SDTM phase?
So we'll have to map those specific data sets to a specific domain as per the SDTM implementation guide. Okay.
Okay.
And we will cover mapping in a detail, right, in the part 2 of the course, where I'll create the DM, AE, and other domains. Okay, I'll give you a detailed description about how we have to map.
Okay. Map, whereas in where we need to map?
Yeah, mapping means creating different domains. For example, A domain, DM domain, CM domain, and then exposure domain, right? So these are called mapping.
Correct.
Yeah, creating different domains as per the requirement.
Okay, fine.
Keeping raw datasets as a source. So here we'll have to use a raw dataset, and then we'll have to map to different domain as per the SDTM implementation guide.
Okay, that I am thinking we will be doing in annotation stage.
No. Annotation is just over there also we are just mapping the variable, but we'll have to collect the data in a specific domain or a specific data sets. For example, all adverse related information we'll have to map in the AE, but we cannot map all variables. Whatever the variable that is mentioned in the AE domain, right, in the SDTM implementation guide, we can only map. So there might be some additional variable, right? So we'll have to map to frequency of AE, and so we'll cover it. Okay, so after that, this is our organization that basically facilitates the acquisition, exchange, submission, and archival of clinical trial data, right? This organization started in 1981, right? And before that, we don't have any standardization. So all the companies let me just go to the next slide. So all the companies here, we have different companies. They have their own standard of capturing the data, and they have to submit the data to the regulator without standardizing. So we don't have standardization earlier, and the regulator has to spend a whole lot of time and resource for validating, right, or for getting the for granting the approval. Okay, so that process is very lengthy, and there is no harmonization. That's why this organization proposes kinds of harmonization at the raw data set level and also at the analysis level. Okay, so here as a programmer, we'll have to use a raw dataset, and we'll have to refer the implementation guideline, SDTM implementation guideline, and we'll have to create the SDTM datasets. This is our responsibility. Once we have a SDTM dataset, we'll have to create the XPT file, and we'll have to submit to the regulator. Yeah, this is also again our responsibility. Sometimes we'll have to create the again, once we have SDTM, we'll have to create the ADaM from the SDTM as a source. Okay, and here we'll have to use ADaM implementation guideline. This ADaM is mainly for analysis purposes, and SDTM is just to separate the data in a specific way. Okay, here we cannot do any kinds of imputation. We can not derive the variables. We'll just have to map the variable from a raw dataset to a SDTM as it is, without any modification, without any derivations. Here we can derive because this is mainly for data analysis perspective. Okay, and here all kinds of derivation, for example, change from baseline, absolute change from baseline, all kinds of derivation, right, we generally do here at the ADaM label. And once we have ADaM, we create a TFSL document, and we'll have to submit those documents, TFSL document, and also the addresses to the regulator. Sometimes in very specific scenario, right, but that is not also recommended. We also create ADaM just keeping the raw datasets as a source, but that process is not recommended because you'll end up with a whole lot of errors, right, and whole lot of queries. But these are maybe sometimes because we don't have proper timeline, right, and we want to do the analysis, So that's why company does, right? But I recommend don't follow that step. Okay, then here once we have SDTM, right, so all the companies has to maybe transform their data into a SDTM structure, and they have to submit those data to the regulator, right? So regulator is now getting a standardized data, and they can very easily review, right, and we can get approval easily compared to a very past where we have to wait for 20 years and 10 years, right, for the from the beginning to end, right now also the drug approval process is around 10 to 12 year from the SS selections to post market assessment, but we can get approval easily, right, because a regulator can approve, or a regulator can review all the data sets quickly because of this standardization. Okay, so they have their own tool. They just have to put the data into the tool, and they can create the report. They can also cross compare the ADaM and SDTM databases in a better way. Okay, and that's why we can generally get accelerated approval nowadays. Okay, because of these techniques. Now I'll describe about the potential of R programming, right? This is very important because here we have all the tools, right? We can do any kinds of sophisticated data analysis. We can do all kinds of sophisticated data visualization part, right? Any kinds of charts we can create, and also we can create a very high definition chart. Okay, that is very important because this ggplot2, this is one of the packages that is very popular, and this makes R very popular as compared to other free software. Okay, so I'll give you a various dimension. We also have a very nice tools, right, for data processing, because as a programmer our 70 to 80% times, right, goes in data cleaning, right? So if we have packages that can clean data in a better way, so it basically save our time, and we can spend our time in other qualitative work. So and also there are other packages. For example, recently they developed one package that is called MMRM that can do mixed modeling, right? If you have if you anyone have some exposure of running proc mix in SAS, so proc mix is very sophisticated procedure. Okay, earlier we don't have any kinds of thing in R, right, that can create a data with that precision, but right now we have MMRM that can create report, right, with the similar kinds of precision, right, same precision. I validated, right, the output of SAS and output of MMRM, and those are exactly same. Okay, so we have such kind of development here in R. So let me describe these aspects. So as of August 2023, R is one of the top 5 programming languages, right, of the year, and it is favorite amongst data analysts and a research programmer. Okay, so this is very important because this is very popular amongst a data analyst and research, because this is free and we have a whole lot of packages, okay, that can do analysis in a better way. And thanks to its veracity, right, because it various industries, right, uses R as a language. Okay, for example, fintech company, academic research, government, right, FT or other weather forecasting service station, retail marketing, social media, right, and healthcare. So we'll discuss healthcare in many more, okay, in detail, right. So it is a platform independent, right? So we can run R on in any operating system. So you can use R on Mac, Linux, right, Windows, and so we don't have to rely on a particular operating system, and that's why this is very cost effective. You don't have to buy a very high configuration laptop. It runs on a normal laptop, right? And it has lots of packages. Right now, as of today, we have, I guess, more than, let me just show you. So we have a repository in R, right? That repository name is CRAN, CRAN. You just have to use that. You have to do a Google search, and you'll get this page, Comprehensive R Archive Network. Okay, you just have to click here. Yeah, I'll show you the installation and download maybe a day after tomorrow. So I'll now show you the list of packages. So here in the software, we have a fourth bullet point, that is packages. Let me open, and as of now we have 21,355 packages. Packages are a group of functions that are clubbed together. Okay, and here you can get the idea about the list of packages published sorted by a data publication. So let me open this. So here this is 18th of September, right? And here we have these many packages published, right? Now we have 8 a.m. right, morning India time, right? And here we have 3 packages published. If you notice the earlier date, the 17th, so these are the packages. Yeah, it's published or some kinds of modifications. And here we have a different kinds of thing, right? Different therapeutic area, sorry, not in clinical domain, other, for example, Amazon Web Service, cost management service. This is the package we can use for that purpose. This programming with big data. Okay, this is a data science kinds of tool, right? And again here, efficient computing for ordinary and generalized Poisson polynomial distribution. This is related to statistics. Okay, so every domain has some presence here. Okay, and we have a whole lot of packages. So we'll utilize the packages, right, that is used in clinical trial settings. And as of now we have 21,000 something packages. Okay, so this basically shows how rich this is. Okay, the next part is here, it is having a various packages for doing a data analysis, right, for statistics, right? So we have here MMRM, we have survival packages, we have packages for summary statistics, we have package HMS that can do missing data imputation, and many more, right? So we have various packages that we can utilize for statistical analysis, and we can do any kinds of sophisticated statistical analysis. Okay, and also in the domain of machine learning and visualization, we have various packages. I will not cover machine learning here. We are trying to create another course on the machine learning aspects. Maybe in coming future we can announce, right? So I'll cover the various packages that is used in a data visualization, right? So I'll cover ggplot2, plotly, yeah, qplot, and then lattice. So those are the packages I'll cover here. And if you ask me about the data wrangling is basically converting unstructured or messy data to a structured one. Okay, and here we have that various packages. For example, we have dplyr, we'll cover dplyr. We have tidyr, we'll cover tidyr also for the transpose purposes. We have also libridate. Okay, for date modification, we'll have stringr for character datasets processing. Yeah, so we'll cover these packages in the class. That package provide a very good functions, right? Whole lot of functions that helps in data processing or data wrangling. Apart from this, R is still growing and it's keep evolving and growing constantly, right? So we have constant update, right? On the new version, they're trying to include various new packages, right, for our request. And okay, so we just have to thanks to the solid community, because we have a whole lot of community that is working right 24/7 just to support us. Okay, we just have to spend some time, we just have to learn those tools and we can utilize for our purposes. Okay, so these are the important feature we have here as compared to the other programming. Okay, and that's all. So this completes the potential of our clinical part. So I'll pause here for the question answer session. I observe that all of you are very silent, right? So maybe you can ask me if you have any question. And don't hesitate. I'm here.
Actually, these things are, Ranjit, like today we have like a session which we are pretty similar, like familiar.
Correct.
So we can ask questions to you.
Okay, perfect.
Once you start our programming, you will get a lot of questions.
Yeah, no worries. I'm happy to answer.
Definitely.
Okay, perfect. Anybody, any question?
Yeah, hi, Ranjit.
Please.
Akshay here.
Yeah, Akshay, please.
Yeah, Ranjit. As compared to SAS, R is more reliable? Like many pharma industries are like moving towards R. Is it so? Like whereas R is an open source, SAS is paid version, right?
Correct.
So is it optimistic to use R in the statistics, like where we work for the most critical data?
Yeah. So we don't have so pre does not mean that anyone can publish anything, right? So they have their own way of validating all the. For example, if you come up with any packages, right, that you want to publish on the CRAN repository that I just showed, right, you have to go through a whole lot of procedure. The testing procedure, your course should be well enough, right? So does not fail, right? You should have to follow certain standard, and they have dedicated team that validate, and they basically allow you to publish on that page. Okay, and also if we so why we have to use this R? So if we use this free software in a clinical trial development, it basically saves the cost of getting a paid software. This is one thing, and they provide additional security, right? Because the companies generally take a cloud version of R. Okay, those are paid, not high subscription, right? So relatively very cheap, right? But they provide additional support, and the companies also have their internal way of validating the packages first, and they only allow the packages that is validated, that is very stable. Okay, that is very important part. Yeah, so they have the internal way of validating all the packages, and then they release list of packages that we can utilize for a particular pharma. Okay, so this is very safe. We don't think that this is open source and this is not reliable as such. No, this is safe. This is very affordable, right? In case of paid, also in the case of cloud version also. And if the company shift to a free software, it basically saves the cost of a subscription, right? Because SaaS has a very large subscription,
high premium.
Yeah, high premium, and it basically benefit the patient. How? Because if the company spends less resource on subscription, our medication become affordable. Okay, so anyhow we are benefiting ourselves at the very end, right? And that should be our noble goal, apart from working as maybe using SAS and R, right? So I'm not I'm not, because every programming has some benefit, right? So we just have to think in that way, right? Well, we can utilize SAS if we can utilize R if we don't have exposure of SAS, right? But
I do completely agree with you, Ranjit. That would be like an interview question. Why do we need to shift to R instead of SAS?
Correct.
Like so I just wanted to be a more particular and keen about to know about this.
No, correct.
Thank you.
No, thanks for asking. Anybody, any questions? Ask anything. So this is very easy programming language. You don't have to face whole lot of challenges. You just have to spend maybe half an hour per day. After the class, you just have to revise the thing what I cover, right? And you are far, right? You can learn this very easily because the syntax are very user friendly, right? You don't have to remember the syntax. You just have to spend half an hour per day, and you are there. Okay, to get all the opportunity. Okay, so any last minute question, thoughts? We have just 3 minutes. Okay, I'll consider silence as no. So don't hesitate, right? I'm here, and if any question is a valid question, right? So you can ask your question, and so we can close the session, right? If you don't have any questions, and we'll meet tomorrow. And tomorrow I'll describe about the historical development of R. Okay, so thank you everyone. Thank you for your time. Bye.
Thanks, Ranjit. Bye.
Bye.
Thank you.